that we're going to talk about. I'm going to introduce optical coherence tomography, or OCT, and then we'll have a lecture on spectroscopic imaging, and then also it's nanophotonic devices. So we'll have this kind of an overview of different methods that you could use in biophotonics. So what I want to talk about with OCT is to introduce some of the concepts, the technology behind OCT, and then go into some different applications. And there we go. I always like to start with this quote because it sort of frames all of the imaging that we do. And it's the front line of research is almost always in a fog. And it's these instruments that function as our eyes, allowing us to gather map points in the fog and to construct a picture of our world in the form of a map or an image, and ultimately to penetrate those obscuring mists. And so if you're looking for a book to read this summer, I would recommend this book by Stephen Hall. And it's quite old. It's probably 15 years old now. But he kind of introduces this idea that imaging can allow us to see things that we can't see any other way, and how it's going to lead us into this millennium. So that's something to consider. Biomedical imaging, of course, is one of these ways that we can peer into these obscuring mists, into tissue and into structures, and we are familiar with all these different instruments. But the focus of this school and this talk is really about optics and how we can use optical imaging in different ways to be able to visualize tissue. And of course, we're interested in many different size scales, like I talked about in the introduction a couple days ago. But with optics, we're afforded this ability to get to smaller and smaller size scales, to look at cells, look at molecular structures, and even vibrational atomic vibrations, and be able to use this information again to detect disease for many applications. And the goal, of course, is that if we can detect disease earlier, we can actually catch it and treat it when it's more amenable to treatment. So that's really a lot of our motivation. Now, if we look at where OCT fits on this size scale, we see in terms of just resolution that OCT is down here with histology on the sort of micron scale. And it just really allows this wealth of information accompanied by many other optical techniques that you've been learning about to be able to visualize structure at this high resolution. So OCT sort of bridges this gap between histology and this really high resolution microscopy with a lot of the more clinical modalities, ultrasound, CT, MRI, and it has many different advantages to kind of fill this gap, which you'll see. Now, it's always difficult to compare modalities. But this is just, I guess, a first stab at that. If we look at resolution, depth of field of view, cost, speed, kind of the capability for sort of endoscopic imaging, you can see where OCT fits relative to, this is multiphoton microscopy. And it does kind of fit in terms of the resolution scale. It's kind of in between these two. In terms of depth, it's intermediate, so there's a lot of advantages. I think that's where OCT in the last 20 years has really come about because it's filled a void that existed. Now, we use light, and I think many of you have seen similar plots like this, where light for us is useful, particularly for a different wavelength range. And so if we look at this range in the near infrared, this biological window of tissue, where absorption is really at a minimum, and the attenuation of light is governed more by scattering than absorption either by water or by hemoglobin or melanin. And this is where we always like to operate, again, for more deep tissue type imaging. It's where OCT is performed. It's also where a lot of diffuse optical tomography and other types of optical methods to allow us to get these photons quite far into tissue. So what I want to talk about today is to introduce the concepts behind OCT, talk about beam delivery instruments to get the light to the tissue, and show examples of morphological or cellular OCT imaging, spectroscopic OCT, and then just a couple applications for imaging cancer and also in primary care imaging. That's going to sort of accompany or lead up to the lecture that I'm going to give next week, which is more focused on contrast agents for OCT and how we can use and design those to complement a lot of the principles that you'll learn about today. So just to lead off, I did send a PDF file to have these notes uploaded. Were they? Yeah. They're available. Great. Okay. So this is just kind of an incomplete history of OCT. There's been a lot that's happened over the last 20 years. It all really started in the 1980s, where telecom was coming about. There were laying fiber optic cable everywhere, and one of the clear needs that was important at the time was to be able to find out, along kilometers, multiple kilometers of fiber, where maybe there is a fault. And so they would send in optical pulses, or they would use this optical frequency domain reflectometry. They would send in an optical pulse. They would look to see, did it come back? And if it did come back, though it would usually come back because of a fault or a splice in the fiber, would send a reflection back. And so they could use this sort of ranging to determine where faults may lie in the fiber. Well, that idea really percolated with Jim Fujimoto at MIT, and they started doing this type of optical ranging in tissue. At first they did this in the time domain, but you can imagine how fast the speed of light moves, and if we're only going a few millimeters in tissue, it's hard to be able to time those reflections that come back. And so really what came out of that effort was the development of OCT. There was a seminal paper in Science in 1991 that talked about this technique using a frequency domain technique where pulses of light are sent into tissue, and using interferometry we can record where those reflections came from and produce like a depth scan into tissue. So OCT was really just taking that depth ranging and making images out of that information. And we'll go into detail about that. Of course, the first application for OCT was in ophthalmology, because of the eye is naturally transparent. And that today is the leading clinical application for OCT. There was also the exploration of using longer wavelengths to image through more highly scattering tissue. There was just a lot of technology development, looking at Doppler OCT, high speed, different applications. In 1997, the first endoscopic OCT in patients was performed. In 98, cellular imaging, improvements in resolution, imaging in the GI tract, the coronary arteries. A lot of contrast agent development came about. And then roughly in 2003, there was a shift in the way that the OCT was performed. And I'll explain all these. We went from a time domain to a spectral domain system. And that really improved SNR. It also improved speed. And that was and that's kind of where we're at today in terms of the technology. Just in recent years, now there's been a lot more effort in just making OCT faster with the GPUs, graphical processing units that have come about, a lot of computational imaging, trying to make quantitative assessments of data, and then just really this rapid commercialization that we kind of see now. So as I mentioned, OCT is about this optical ranging in biological tissue. So it's a lot like ultrasound, where we're familiar, which I think many of us are more familiar with. We send in ultrasound, you send in sound waves, and you collect the reflections that come back. The speed of sound is quite slow relative to light in tissue, and so therefore you can use electronics to sort of gate out when those reflections come back and figure out where that reflection occurred. With light, however, it's too fast, right? So we can't use just gating electronics. We have to use another technique, interferometry, to detect where those come from. So to understand how we would use interferometry, we have to go back and look at coherence theory, okay? So what does it mean for light to be coherent, okay? And in that sense, what does it mean for light to be low coherence? So if we look at this temporal coherence function, temporal meaning it's in time, and we say that this U of t is a stationary complex random function, the temporal coherence function is defined in this manner. And you can see that it's simply the U of t, the complex conjugate, times the same function but delayed in some time tau. Because this is the same function, it's often called the autocorrelation function. So it's how the function is related to itself at some delay in time. Okay? So this g of tau carries information about the coherence of that function. Okay? And we can normalize that by just looking at what is that function without any delay, tau, where tau is 0. And we can normalize that, and we get this small g of tau, which is this complex degree of temporal coherence. And it's going to vary between 0 and 1, where 0 means that these two functions are completely uncorrelated. There is no relationship between the two. Whereas 1 means that they're perfectly correlated with one another. And so generally this is going to fall in between these values. Now we can also derive from this, and I won't go into all the math, but we can also derive a coherence time, meaning if a wave originates from one point, how long does it take for that wave to sort of lose its relationship with the original source that it's coming from, okay? This is the coherence time. There's also a coherence length, which is the coherence time times the speed of light. So over what distance does that light have to travel before it becomes less correlated with its source, all right? And there's also the spectral width. We can see that there's a relationship between this correlation, this coherence time, and the spectral width. Now, there is a Wiener-Kitchum theorem that relates the autocorrelation function, or this coherence function, with the spectrum, S of nu. And this is a really important relationship, and this is one that, for people that have done the OCT lab, this is what we highlight in the lab, because this is really a fundamental basis for understanding not only OCT, but just coherence properties of light in general. So there's this relationship. This S of nu is the power spectral density, okay, of the light. Let's look at this graphically. So these are our autocorrelation functions. So in a sense, it's telling us over what time are these two waves correlated, okay? And how are they related to the spectral density function, or the spectrum, the bandwidth of that light? We can see that there's a Fourier transform relationship between these two. So what that means, as I remember, is that if something is narrow in one domain, the time domain, it's going to be broad in the other domain, in the frequency domain. So if we have an autocorrelation function that's really narrow, okay, so that means the light is only going to be correlated over a very short period of time or space, then its spectrum is going to be broad. We want to, these broad bandwidth sources give a very short coherence length. And similarly, if we have a very narrow spectrum, like a laser, a standard laser that has a spectral line width of maybe less than a nanometer, then it's going to be correlated, that light is going to be correlated over a long distance in time or space. Okay, so that's this relationship. When you look at the bandwidth of the light, if it's a narrow bandwidth laser, it's going to have a very long coherence length, okay, or long coherence time. If it has a broad spectral bandwidth, like a LED or a superluminescent diode, or even the sun, right, it's going to have a very short coherence time or length. It's that inverse relationship. So how do we use this with OCT? Well, we use interferometry to detect where those reflections come from. And we have our light source. We split the beam into two, a reference arm and a sample arm. And we're going to get reflections that come back from the reference and the sample arm. They're going to be recombined and then detected at our detector. Now, our reference in this case is a mirror that's going to be moving back and forth. So it's varying the path length of the light in that arm, and it's also varying the time at which it's going to take for that light to come back. Now, if we have a source, like a laser, a standard laser that has that long coherence length, right, the interference we're going to see at this detector is going to be over a large displacement of that mirror. We're going to see our interference fringes, but we're seeing that the amplitude of this is fairly constant. That's because it's coherent no matter where or how much we vary that delta M. And that's the case where the coherence length is much, much greater than that change in the path length, delta M. If we have a short coherence length light source, like an LED or a superluminescent diode, then there's only going to be a limited distance over which we're going to see these fringes. Because as this reference arm mirror is moved, okay, the light coming back from the reference arm may not be correlated anymore or coherent with the light coming back from the sample. So as we sweep this mirror, we're changing our delta m, or we're changing the z, and we're going to see that we'll trace out an autocorrelation function that looks like this. And the width of this is that coherence length of that source. Okay, is that clear in terms of how we choose our sources? What this allows us to do is if we put in a long coherence length source, we can't figure out where that reflection came from because the signal amplitude is the same no matter what position this mirror is. But if we use a short coherence length light source, then as this path length changes, we're going to localize in depth where our signal is coming from. And the amplitude of this is going to tell us how strong is that reflection at that one particular point. Okay? Here's a little bit more of the detail. So now we're performing interferometry with a low coherence light source, and we have two partially coherent waves, U1 and U2, and they have given intensities. The cross-correlation function, this G1 and 2, is defined this way. So now it's not necessarily the autocorrelation function, but we have two separate waves. We can normalize it, and we can generate this interference equation, which is as the intensity that's measured at our detector. That intensity is the intensity I1, I2 coming from these two waves, but there's also this term. And this is somewhat of a DC term, it's constant. But this term is going to vary. And it's going to vary by this little g of 102, that autocorrelation, that correlation function. And it's also going to vary by this cosine of phi, which is phase difference between the two arms. Okay, so this is really an AC term, and that's what's going to trace out these fringes. Okay, we can look at the amplitude of that. That amplitude is really governed by the coherence properties of the source. Okay, and this axis, the x-axis, is governed. It's the temporal delay. It's really the difference in path length between the two arms. Okay. All right. So that's how we get these autocorrelation functions. If we put a mirror in our sample arm, we essentially will generate one of these little autocorrelation functions off of that mirror. And you can also see, hopefully, that the width of this not only defines that coherence length, but it also defines our axial point spread function, okay, or resolution. So this is our standard beam focusing. If we have a low numerical aperture, we know has a long depth of field. Okay, that's B, our confocal parameter. Our width here is 2 omega naught. Okay, so this is really defining our transverse resolution. Our axial resolution, though, is defined by that coherence length. And so as we move that reference mirror in different positions, we're essentially moving the position where we can collect signal coherently. Okay, so we can, if that reference path is far, we're going to be measuring down here. And the amplitude of that envelope is the intensity of the scattering that's coming back. Okay, so as we move that reference mirror, we're moving this sort of window or this coherence gate in depth. Now, if we have a high NA optic system, then we know that the confocal parameter is much reduced, so the depth of field is shorter. The transverse resolution is smaller, which is better for higher resolution. But we also have to make sure that our coherence gate overlaps with that small volume. This is how we do optical coherence microscopy, which is more for en face imaging, whereas this is standard OCT, where we're doing depth resolved cross-sectional imaging. So to do this then, if we move that reference arm mirror in depth, we, at one position, we're going to collect, this is the envelope of that interferogram. And the intensity corresponds to the amount of optical backscatter that's coming back. That's a single column. And then to produce an image, we move our beam over and we repeatedly collect these columns and we assemble our two-dimensional image. Two-dimensional cross-sectional image. This can also be three-dimensional in the other, if we add the other dimension. That en face imaging is what we would use for optical coherence microscopy. Okay. Well, a lot of people have done, I guess, any questions about more of the coherence theory, the theory behind how we're going to generate OCT images? Okay, stop me. Sometimes I tend to go pretty fast. There's been a lot of work by other groups that have explored the properties of light and interacting with tissue. This is by Wolfgang Drexler's group, where he looked at different properties of tissue for different wavelengths. And so this is at 800 nanometers, 1060, 1300, all within our biological window. But you can see at different layers within the skin, we see very different contrast. And this is all because of the different types of absorption or scattering properties of the tissues, the cells at these different layers. So a lot of this is useful for imaging particular tissue types. We generally know that scattering in tissue is decreasing by 1 over lambda to the 4th. And so we tend to want to use longer wavelength light if we want to image deeper into tissue. Of course, though, if we use longer wavelengths of light, you can see it, we do image deeper here in skin compared to 800 nanometer. But we do sacrifice some resolution by using longer wavelengths of light. This is another example of this, where using the longer 1300 nanometer light, you can see there's a bit more structures that can be resolved down here. This is also a nice example of how the different bandwidths of the spectral bandwidths relate to these autocorrelation functions. And you can see that we have a very large bandwidth, 470 nanometers here, which is going to give us a narrower coherence length or autocorrelation function. This is sort of experimental data that reflects that point. So in OCT, we always tend to want to use a broader bandwidth of light to get higher axial resolution. But there's all kinds of other inherent problems that if we have a non-Gaussian looking spectrum, we tend to get side lobes in our autocorrelation function. And sometimes if those side lobes are too large, then you actually worsen your resolution. So these are some of the nuances, but things that we have to be aware of. It's a very good exercise just in MATLAB to actually define kind of a spectrum and then take the Fourier transform or inverse Fourier transform of that and look for sort of this autocorrelation function and go back and forth. Okay, so what I was describing was really time domain OCT. That's in the case where we have our broadband source, we have a reference arm that's moving back and forth, and this is really where OCT started. We would get depth dependent or time dependent data. We would take the envelope of that, and that's where we would get the single depth scan in Z. Well, in 2003, like I said, it really came about there's other ways to collect this data. And one of those ways is called spectral domain OCT. The other is swept source OCT. Now, in spectral domain OCT, in both of these, what happens is we're going to collect light sort of in this Fourier domain. We're going to collect spectral data. We're going to then inverse Fourier transform the spectral data to get data that's in time or in depth. And we can use the positive portion here, which essentially is the same information we collected with the time domain OCT. So what's different? Well, in spectral domain OCT, we use the same broadband source, but you'll see we have no moving reference mirror. So instead, what we do is we collect light, we replace our single photodetector with now an array of photodetectors, or a line scan camera. We take a grating to disperse the spectrum across that camera, and what we're going to see is fringe data across the line scan camera. So we're essentially collecting backscattered light from all depths along that column at the same time, and we are encoding depth by frequency. In the case of swept source OCT, we go back to a standard single photodetector like we did in the time domain. But instead of our broadband source, we have a narrowband source that's rapidly swept across many wavelengths. And as the swept source is swept across, we trace out this fringe data over time. So this detector is reading out signals that still look like this, and then we can use our same processing. The advantages, of course, are manyfold, but both of these are faster, can be faster than time domain. In the spectral domain, we have no moving parts, which is a huge bonus if we're trying to maintain sort of phase stability of our light. In swept source, this is still mechanically swept in some form. We're mechanically sweeping that laser. And so this can induce some instability. The advantage of swept source is that this is a laser now. It's not a superluminescent diode. So we can get a lot of power out of a laser. And so this tends to give us better signal to noise as we go faster and faster with acquisition rates. All right. So how does OCT compare to confocal? Many of you are probably familiar with confocal microscopy, where we have a point source of illumination that's focused to a focal plane, and we have a confocal pinhole in front of a detector that is essentially only allowing light that's coming back from the same focal plane to be detected. So this is really spatial rejection of light. Any light that's above or below that focal plane is going to be spatially rejected by that pinhole. OCT, in a sense, operates like a confocal microscope because we generally have optical fibers that act as pinholes. So to get light back into an optical fiber, it has to be in this single mode. It has to be coming from that focal plane. But in addition, we have this coherent rejection. So just as I said, we rely on light that's backscattered from the sample and recollected. Any light that gets scattered around in the tissue that may still happen to get collated by our system is rejected, because when it bounces around, it loses its coherence. It's no longer coherently related to the light coming back in the reference mirror. So we have both spatial and coherent rejection of light. And that's why we can image with OCT millimeters into skin or highly scattering tissue. Whereas with confocal, you're lucky to go 50 microns. And it's because of this coherent rejection. Okay, well, what about the bandwidth? And how does that relate, again, to axial resolution? This is our standard relationship. The coherence length, you can see, is inversely proportional to the bandwidth of the source and related by the square of the center wavelength. So if we just look at 800 nanometer light from a Ti sapphire, we can see that as our bandwidth increases, our axial resolution or coherence length will decrease. So we really have to dramatically increase the bandwidth way out here to 200 nanometers or so to get down to micron resolution. And that's always been a challenge. This, of course, is also just assuming we have a standard Gaussian spectrum. Here's an example of comparing a superluminescent diode bandwidth of about 32 nanometers to that of a Ti sapphire that has really exceptionally broad 260 nanometer bandwidth. And if we look at those autocorrelation traces, we see the SLD has a spectrum that's or a coherence length of about 11 microns, and then of course its autocorrelation looks like this. We can look at then the Ti sapphire. It has a much narrower autocorrelation function. You can see there's side lobes, but its point spread function is quite narrow, so 1.5 micron. So we always want to try to achieve this broader bandwidth. Now to get there, you've probably heard a lot about supercontinuum generating fibers. And this is one of those methods that allows us to generate that broad bandwidth. We essentially will pump these fibers that have a very small core surrounded by essentially air, and we can get this nonlinear broadening. So we can have a very small, a narrow pump, which is this dotted line here, and generate very broad supercontinuum. This happens to be quite noisy because this supercontinuum is generated by nonlinear processes in the fiber. And so while this has been great for bandwidth, it is not always the best source because it's a noisy source. Some of the work in my own group, we've been looking at trying to broaden these the supercontinuum, but then also correct the phase of the light. One of the things they don't tell you, if you look at a lot of the supercontinuum, we see a nice broad spectrum, but that broad spectrum has come about because of millions and millions of pulses. And this is the average spectrum, but a single shot, a single pulse, the spectrum from a single pulse is quite spiky. And what that means is that the light down here at 400 nanometers is not necessarily coherent with the light up around 1500 nanometers. So a lot of the work in my group has been to try to ensure that we have coherence of all the wavelengths and photons across that entire supercontinuum. And so there's just a lot here. I'll just refer you to some of these references. But what this is allowing us to do, we're actually adding a pulse shaper to shape the pulses in time to correct this phase. And we've been able to compress these pulses down to extremely narrow. This shows 10. We've been down to about 5 femtoseconds. So if you can compress a pulse down to a very short duration, that means it's got to be coherent over a much broader bandwidth. Okay. There's a lot of other advantages we get from these fibers. One thing that we can generate is what's called Cherenkov radiation. And so by choosing where we pump our fibers relative to some of the fiber properties, we can get tunable wavelengths over the visible spectrum, even into the UV. So we have a lot of options for being able to design sources for OCT, either in the visible wavelengths or out here in the biological window. Some of this work is also relevant to our nonlinear optics, CARS, and other techniques where we need these different bandwidths and different center wavelengths. Okay, so in general, this is how we'll put an OCT system together. It's fiber optic based. We have a sample arm, a reference arm. If we're doing time domain, we have a Galvo. If we have spectral domain, we've got a line scan camera. And we can get typical resolutions down the 1 to 2 microns or so. We can get a lot of power out of these sources depending on the sources we use. But we can do real-time volumetric imaging with these systems. This is just some experimental data that I wanted to show you what this spectrum looks like when it's displayed on our line scan camera with spectral domain OCM. And you can see this is the outline of the spectrum, but it's modulated. That's the interference fringes coming from all the different reflections in depth. Okay. So when we build these systems, fortunately, a lot of the hardware is based on the telecom industry. And so we can get very small sources, detectors, fiber optic components. A lot of the engineering is going into the probe system. So how do we deliver the light to the tissue where we want to image? We can integrate this with surgical microscopes or catheters, fiber optic catheters, needle biopsy probes, handheld probes, and all these can be on a very small, the OCT system can be on a small cart. And we'll continue to see these systems get smaller and smaller and more portable. This is just an example of one of these catheters where we use a single mode fiber. We have this is a Grin lens and a prism to direct the light out at a radio direction, and these little micro optics are spun inside the catheter. And these catheters can be inserted into endoscopes and be able to access internal body sites. These needle systems, we can design a single fiber, a focusing lens, a Grin lens. Maybe we can angle that to direct out to the side, but it allows us to collect, in this case, single axial scans from different tissues. And we've used this in breast tissue to look at differences between adipose, tumor, and stroma. And you can see there's clear depth-dependent differences in scattering. We can take these same needles and make measurements or design needles to measure refractive index. The refractive index is going to be based on the differences that we see in the scattering at the tips of these needles. So there's a lot of different designs that we've built where, in this case, we have a fiber and then we have the light going across this little channel, and this channel is where tissue may be resting after we insert the needle. And we can actually measure the change in path length across this channel, and that change in path length is going to be proportional to the index of the tissue that's lying there. We can take these needles and also mechanically spin them or translate them, so we can get small cylindrical volumes of data based on OCT. So a lot we can do with these needles. The more recent work that we've done is to take this type of cylindrical volume imaging and put it into these larger core needles. when we have to sample tissue, like a lesion in the breast, one of the ways is to insert a core needle biopsy system, which will go in and actually take a small core of tissue that preserves the structure. And the pathologist then looks at the structure of that little core of tissue. Well, up till now, all the use of these OCT systems and needles have always been to look at the tissue, not really to biopsy it. And so this is a system that we modified, a commercial system, so we can do OCT imaging immediately prior to taking that physical biopsy. We can also use to guide the placement. So this is the standard biopsy needle, and what we did is we added a tube that runs the entire length. We opened up the tip and added some transparent plastic windows so we can do our OCT imaging at our tip. And this is just kind of a blown up area where this is the transparent tube where an OCT catheter is inserted. The catheter can be in this front window to guide the position of the tip. We can then pull the catheter back here, and this is the channel where the biopsy, the tissue, is actually cut out and taken. And so we can actually image that tissue immediately before and after we take the physical biopsy. So these are just some examples. As we're inserting this needle in, we can see areas of normal tissue, and then we kind of hit areas that are tumor, and we can then guide where this is located. This is actually tissue that's been retained in the biopsy needle. So you can see after the tissue has been excised, you see this continuous scattering from the metal needle barrel, and we can see the tissue and evaluate the tissue once it's been biopsied. Of course, this is the histology as well. These are just some examples, but OCT really has just taken off commercially in the last couple years. There's probably now 20 or so companies and at least that many product vendors that are coming together to build these types of systems. And you can see they're all taking various forms of carts with different modules to look at the eye, to look at the skin, interoperative use. It's really taken off for many different commercial applications. So just to kind of summarize this technology part, again, with OCT, we want to use the near-infrared, the biological window. We can have very high resolution, high speed acquisition, even faster than this now. Imaging down to a few millimeters, which is better than any other sort of optical microscopy system. And we can integrate these fiber optics with many different beam delivery systems and have many other types of accessory adjunct techniques like Doppler and spectroscopic. Okay, so really when we think about using OCT, what's really going to be needed to do this type of diagnostics, right? And we can use OCT to look at structure. All the images I've been showing have been mostly structural. And that's the morphological imaging. We ultimately want to try to improve resolution to get down to cells and ultimately even to do molecular type imaging with contrast agents or other just endogenous techniques. So if we look at just some quick examples of morphological imaging, as I said, ophthalmology was the leading application. This is an example of the optic disc and fovea of a human. And you can see there's, with this high resolution imaging, we can start to see individual retinal layers. We can really define structural changes that may go on. And this has really advanced dramatically over the last 10, 20 years, where now comprehensive 3D imaging is performed in real time, and all these individual layers within the retina can be assessed. So changes of retinal pathology can very easily be detected, and I could almost guarantee everybody in this room will have an OCT of their eye sometime in their lifetime, if not already, because it's just proliferating and moving from the ophthalmologists to optometrists, and also, as you'll see, maybe to even a primary care. This is some examples of structural imaging in the GI tract. So this is the esophagus. You can see that we can image about 1 to 2 millimeters deep, and this is the corresponding histology. We can see a lot of detailed structure of the colon, and this is a condition, colon pseudomembrane, where it's really kind of broken up, and you can see that OCT gives us a pretty good representation of what the histology, would show if you were to take the tissue out. Catheter-based imaging was used to look at Barrett's esophagus. This is a condition of, if anybody has heartburn, you can get acid reflux into your esophagus, and that changes the cells in your esophagus, and you're at risk for cancer. But they do this type of surveillance, and we can see, this is pretty obvious where it's on the surface, but we can see structural changes, this glandular changes in the mucosa, which indicates this condition. So we can use, again, to look at the structure with OCT. Cardiovascular imaging is really becoming more and more common these days with OCT. And this is just a comparison of the resolution we get with OCT compared to intravascular ultrasound. So roughly about a 1 to 2 order of magnitude higher resolution than ultrasound. And so we can begin to make out these individual layers. We can look for plaques and assess whether or not those are prone to rupture. Looking at the skin, we can see a lot of structure, the sweat glands. We can see down to the dermis, epidermis junction. If we use optical coherence microscopy, we can really start to see individual cells. And this is some of the work from my group, that we can use both OCM and multiphoton. This is 2-photon fluorescence, autofluorescence from the skin at different depths. And it may be hard to see, but every one of these little dark specks are nuclei. And down in the superficial dermis, these are the little papillae that are coming up between the dermis and the epidermis. And so we get exceptional resolution, and we can image quite deep with OCM than we would with confocal. There's a lot of work on cancer imaging. I'll talk more about that in a second. But this is just an example of a very high resolution image, structural image of a tumor from a dog muscle. And you can see the scale here. This tumor is about the size of the tip of your pen, extremely small. But we can clearly see structural differences between normal and abnormal. OCT has been used in real time because it's fast to track things such as surgical interventions. This is an example of liver that is being heated for hyperthermia treatments. And you can see optical changes that are happening. And in this case, the laser was stopped before there was any significant mechanical effects, and we can see a small lesion on histology that was formed. If this goes too far, then we get this mechanical tissue disruption and very gross structural changes, mechanical changes. So in this sense, OCT can be used to maybe control the administration of these types of therapies just by looking at the structural images. And this is a little bit more subtle difference. There's a condition called port wine stains, which are hypervascular added capillaries close to the surface of the skin, so they leave these red lesions. Well, it's often used, visible lasers are often used to go in and try to coagulate those blood vessels. And in this case, you can see there's 2 small regions. This is kind of a better region here, where there's small blood vessels about 1 millimeter below the surface of the skin. And we're applying a visible laser to heat the blood in these blood vessels. And it may be subtle, but you can see that these vessels tend to be closed off compared to these open lumens here. And if we let this light continue, we start to see injury of the superficial skin. But if we look at the histology, indeed what has happened is that the blood in those vessels have been coagulated. So we can use, again, in this OCT as an example to guide this type of coagulation and use this as a sort of feedback as to how much light or dose to deliver. Okay. Any questions? I have to catch my breath, but any questions? All right. A lot of material, and I'm trying to get done in a reasonable time. Cellular imaging. So we're pushing this limit now. Why did this die? There we go. This tadpole, Xenophus laevis, has been kind of our friend for OCT for 15 years. It's an ideal specimen to use. These are tadpoles from this African frog. That's a really common developmental biology animal model. And what's nice about it is that they develop rapidly. They're semi-transparent. We can see all kinds of interesting structures. And so we can see, start to see individual cells in these in these specimens. So we can use OCT to look at three-dimensional volumes of these cells in this gill-like structure. We can also temporally acquire the beating heart from these tadpoles, and we can track these individual cells as they're growing, dividing. And this is the corresponding histology, which matches quite well. So this was a really early study I did during my graduate years, where I was able to identify cells, a parent cell, and actually watch these cells divide over time. These are images over the course of several hours. We could also track cells that are moving. So there are a set of stem cells that originate from this neural tube and will migrate through these developing organisms. And they happen to be filled with melanin, so we could track where that cell is located as it's moving through the tissue, and develop three-dimensional maps to understand how these cells are moving through tissue. This was again done over a decade ago. Where we're at now is we can use not only OCT or OCM, but combine with multiple modalities to track how cells are moving within tissue. So we can use OCT to look at scattering, also phase changes, which indicate blood flow. We can combine our systems and a microscope to do multiphoton microscopy or fluorescence lifetime imaging, and we can start to see these really fascinating combined structures or images of all these structures. So the gray signal here is actually the capillary network. We're looking at skin, and we're looking at skin from mice that have had a bone marrow transplant. So we've taken GFP cells from one mouse, we've eradicated the bone marrow of another mouse, and we've transplanted the bone marrow. So any of the green cells that we see in this images are GFP cells that have come from the bone marrow of this mouse. And we're using this to track where these bone marrow-derived cells are moving. So we can see really intricate relationships. In the skin, the black areas are the follicles. The capillaries surround all those follicles. We actually can wound the skin with a really small pinprick, and we can see how these cells are all migrating around the wound to repair that wound. This is a time-lapse sequence that shows the OCT of the follicles. It shows the capillary network, and then it shows those GFP cells and the hair shafts over the course of 35 days. And these are all perfectly co-registered, so we can see how these GFP cells come into the wound. We can see how after the wound is placed, how we start to see angiogenesis, and the vessels are recombining, and just really a wealth of information. Yeah.
If you're doing it over a period of 10 days, do you not get photo bleach?
So we do this, it's actually over 35 days, even longer. Ben Graff, who worked on this project, essentially would bring the animal in, would do imaging, and then imaging perhaps maybe an hour or so, and then the mouse would go back to the cage and be brought back and forth. And he developed a pretty sophisticated registration algorithm that would allow, even though you bring the mouse back, days later, you can still find the same location based on where the hair follicles are located, and have this type of registration. So in terms of bleaching, I don't think he observed any bleaching of the GFP over that time, but the exposure times were probably only minutes at a time. So he would take the images and then obviously block the beam, and the mouse would go back. So we didn't see any of that GFP bleaching. Yeah, there's it's hard to say because these cells are coming in, so maybe some of these cells that were imaged before had been bleached, and these are new cells coming in. The real bright collection that you see in the middle on there is actually the scab of the wound that then falls off naturally. And the reason it's so bright with GFP is that these cells had come in, they had died, and formed the scab. And so that GFP, that fluorescent protein, is still in the scab itself, but then it's lost. So this is really, this work was just submitted for publication, but we there's a lot of information here that we have to go back and analyze. Okay. One technique that is sort of an adjunct technique that comes out of this relationship between the spectrum and the envelope of the OCT data is called spectroscopic OCT. And if we look at the envelope of this axial scan, that really gives us a structural information. But if we look at the carrier frequencies, the interferogram, that can tell us something about the spectral reflection, the scattering, the absorption properties of the light and how that passed through the tissue. And that's called spectroscopic OCT. Here's an example. So if we take our Ti-Sapphire laser that has a spectrum that looks like this, and it's sent to a cuvette that contains a dye that has a center wavelength absorption at 740 and has a 10 nanometer absorption spectrum. We look at the reflection coming back from the front surface. We see the autocorrelation function. We take the Fourier transform and we get the spectrum. That looks like the incident spectrum. If we look at the reflection that comes back from this back surface after it's gone through the dye, we see this distorted autocorrelation function. And that's because the dye has absorbed some of the light, some of the wavelengths of that incident spectrum. If we then take the Fourier transform of that autocorrelation function, we get back this spectrum, which you can see has a notch at 740. And that's because of the sharp absorption of that dye. So this information that we get back, this is experimental data. This data that we get back from these reflections, we can use that information to tell us about the absorption or spectral scattering properties of our sample. Now this is called spectroscopic OCT. It can be done, again, in both the time domain or the frequency domain type systems. Either way, we can get access to the spectral information and we can recover that. And so to do spectral OCT, we tend to look at the centroid of the spectrum, and we can tell, has that centroid shifted? Has the dye or has the tissue absorbed certain wavelengths of light within that spectrum, and has it caused the centroid of our laser spectrum to shift? And we can assign a color map to that shift. And so that's what's done here. This is a structural image of a tadpole. This is Barrett's esophagus. And if we look at the spectroscopic information, we can see that the color scale here is if the centroid shifts to longer wavelengths, it's red. If it shifts to shorter wavelengths, it's green. And we can see things like these melanocytes caused a very dramatic absorption in the shorter wavelengths, which resulted in a shift to the longer wavelengths of the centroid. In the structural image of Barrett's esophagus, it's somewhat difficult to tell the difference between these two types of epithelium. But spectroscopically, we can see that there's a more distinct difference. So it's this type of spectroscopic analysis that can give us more information about the tissue. Okay. Well, there's other ways that we can deal with this. This technique is not all that easy because there's a lot of things that can shift this spectrum. We built a multiphoton, an integrated microscope that combines OCM and multiphoton, and I showed you some of this data already. But if we look spectroscopically, we can collect images with this microscope of multiphoton, of OCM, but we can also, I'm sorry, I'm kind of jumping around here. Let me find my point. Okay. I think let me start here. So we want to use this microscope to be able to detect these spectral shifts. And as one example of this, we can take an agarose dye, an agarose sample, and we can load it with the dye. And we can have also another sample that just has agar. And we can see that with the dye, as we go deeper and deeper, we're getting absorption at the shorter wavelengths of our spectrum. And therefore, the light we get back at deeper depths has been shifted to longer wavelengths. Okay, and we don't see that shift without the dye. So this is a way, again, of sampling how is the spectrum changing in different depths. Now, we can use that same type of an approach to try to characterize even individual cells. And I'm sorry that this didn't come out very well, but we seeded cells into a gel, different types of cells, macrophages and fibroblasts, and we can see that each of them have a different spectral shift. This one looks more yellow in appearance. This one looks more purple in appearance, and it's because the spectrum we're getting back has been shifted. So we've explored a lot of different ways of using spectroscopic OCT to try to differentiate tissue structures. Now, one of the trade-offs that may be obvious to you or may not is that we have to constantly trade off a time-frequency resolution. If we think about our interferogram over depth, we know that if we need to take a section of this in order to do the Fourier transform and recover the spectrum. If we have a larger area, then we get better spectral resolution, but lower spatial resolution. And so this is that inherent trade-off. If we want to be able to recover that spectrum and analyze it, then we have to sometimes sacrifice spatial resolution. We can get around this if we use our high numerical aperture system, because we know spatially where the signal is coming from. Okay, it's no longer depth resolved, but we can confine it to this volume, and then we can do the spectral analysis over our large interferogram signal. Okay, when we do this, these are the types of spectral enhancements we can get. This is looking at rat mammary, adipose, and muscle tissue. And this is the OCM image. This is the histology. But we can look at how the spectrum shifts to provide us with different types of contrast. In this case, we're looking at perhaps where nuclei, very dense structures of the muscle and the nuclei are located compared to this adipose tissue. And these are just, again, some of the methods that we can use. My batteries should go in here. Okay, if we do this on single cells, this sort of proves that point. This is a cell culture using our integrated microscope. These are green fluorescent protein fibroblasts that have been co-stained with the nuclear dye. And so the nuclei all show up as red. And we get this multiphoton image, we get our OCM image, and we can overlay these channels. When we do our spectroscopic analysis, what we're looking for is how have scatterers shifted the wavelengths of the light. And we can see that we see these bright little peaks that happen to correspond to each one of the nuclei. So it's these nuclei that are spectrally shifting the light that we get back. And we can detect that spectroscopically with this method. Okay. Well, I just want to end by a couple examples. Cancer is one of those. We all know the impact that cancer has had on our society, on our lives. We know diagnosis is important, early detection and diagnosis. And we want to be able to develop imaging systems to be able to help us with that early detection. Now, the process of cancer, as Marina talked about as well, follows a pretty prescribed sequence of dysplasia, sometimes in the case of epithelial cancers, carcinoma in situ, and then malignant cancer where the cells have burst through this basement membrane and have started to metastasize through the bloodstream. There's lots of points where we can actually look at morphological changes, but we want to get to the point where we're moving from morphology to molecules and move from histology to in vivo type imaging. So what we want to do in this case, what we've focused on, is how we can change the paradigm for when a diagnosis or assessment, microscopic assessment of tissue is made. Traditionally, that's made in the path lab on samples that are taken out. And we want to do that in real time in surgery or at the point of care. And so a lot of studies have been focused on breast cancer. And early on, we noticed how OCT images can look a lot like the histology. They don't have the same resolution as optical histology does. They don't have necessarily the selective staining that histology does. But we can get this information in real time at the point of care. And sometimes that's really important. We also notice in lymph nodes that there are structural changes in lymph nodes when they have tumor cells or reactive changes that show up pretty distinctly on OCT compared to the normal condition. So can OCT actually be able to assess lymph nodes and how tumors may have spread? So we have still even today ongoing research where we take OCT into the operating room. We image tumor margins from breast lumpectomy specimens. We also then compare we mark areas and we compare our OCT images with the histology that's coming back. And we see examples like this where if there is a negative margin, we see mostly fatty tissue, adipose tissue. If we start to see focal areas of highly scattering cells, that's indicative of tumor cells that have been left at the margin, meaning that there's likely tumor left behind. And we can provide this information in real time during the operating procedure. This is one last example of a really small collection, less than 1 millimeter collection of tumor cells and the corresponding histology, something that would not be visualized by the surgeon intraoperatively. So we can, of course, do this in 3 dimensions and collect these this data for analysis. And these are examples of lymph nodes where we can see a lot of the internal structures of lymph nodes and determine whether or not tumor has spread to these nodes. This was a couple years ago, but we finished a study of about 38 patients which showed a sensitivity of 100%, specificity of 82%, of finding tumor in the operating room, tumor at the margin, as opposed to in the pathology lab. Yes?
I have a question.
Yep.
Do the pathologists have a special training to interpret the images?
Yes.
Do the surgeons, are they able to understand? Or
that's a good question. Yeah. We, in this case, the person making the call on the OCT images was a grad student. So someone trained in OCT. But we imagine that a surgeon, it would be the surgeon who's likely going to be the one interpreting real-time OCT to say, is this normal or abnormal? We also envision, just like they do for frozen section histology, where they'll have a pathologist waiting outside the operating room to look at tissue, that pathologist may be looking at these OCT images in real time as well to help with that diagnosis. So we're moving in this direction. We have commercializing these systems to do this intraoperative imaging, but these questions have really always come up: is who's going to see the data, who's going to interpret it, who's going to get paid, for these procedures. They all want to know that too. Yeah.
So for high sensitivity, is not possible to develop some algorithm for automatic notification?
Exactly. Yeah, and we've done some of that. So as OCT has gotten so fast and can collect volumes of data faster than the human eye can interpret, so we have some algorithms we've started to develop that will look at the amount of scattering, look at the spatial frequency components in the axial scan, and can assign, flag areas that look suspicious. So that's what we.
It's not that easy, but in the case of such high sensitivity.
Exactly. Yeah. So those are areas that I think are going to be important as well. And this is an example of I have a spin-off company that has now a handheld probe that's allowing us to actually do OCT in the tumor cavity in the patient, not just on the resected sample. And this is, I think, it's more important to determine what you may have left behind as opposed to what you've taken out. I know Professor Carney probably talked a little bit about the ISAM work that we're doing collectively, so I won't go into detail. But there's a lot of other techniques to improve the resolution for OCT, and we're applying that to our intraoperative use. Stephen Oddie, who you're going to meet in the OCT lab, worked on using a computational method to correct aberrations in these optical systems. And so this just came out in PNAS. But what it shows, it's a computational way using OCT data to correct aberrations in the system. And so if you watch, this is the focal plane. By computationally adjusting the parameters in the collected data, we can bring that data back into focus. So again, this is all coming into focus. These are other planes that are being corrected as well. So there's a lot of, as I said, there's going to be a lot more work on developing computational techniques for tissue identification, for aberration correction. Okay, the last point I want to make is that if we look at our healthcare system, I tend to break it down into these areas. The primary care, the doctor who you see first for any issues, or maybe you see the emergency room first. Hopefully you don't go to the operating room, and maybe hopefully you don't have to see a specialist. But these comprise our system, and at least for OCT, a lot of technology is geared at the specialist in ophthalmology and cardiology. But we recognize that much of the screening that can be done, much of our interactions with the healthcare system are with the primary care office. And if we just look, in every one of these offices is an ophthalmoscope to look in the eye, or an otoscope to look in the ear. And so we've developed OCT systems that really can advance these systems at the front line and be able to collect 3D OCT data of the ear, of the eye, and trying to get this in the hands of the primary care physician. So they're the ones that are going to be seeing the general population and potentially could act as better ways to screen for early detection of disease. And this is just some examples of this handheld system. This is the oral mucosa looking for oral cancer, the skin, the cornea, the tympanic membrane for ear infections, the fovea, and the optic disc. And all this with a handheld MEMS-based scanner. So just to wrap things up, I know I went through a lot of this quickly, but OCT I think is really become robust, versatile, is expanding rapidly. It's great for morphological, cellular, potentially molecular imaging, and in many areas of healthcare, we're starting to see this impact. I've shown some examples of image-guided biopsy, tumor margin assessment, primary care imaging, and the list really just keeps growing every day. So I talked about these things today. Next week, again, I'm going to focus more on the contrast agents and new contrast agents that will help with enhancing contrast or site specificity when using OCT, and we'll talk about that then. I do want to thank everybody from my group, and as well as our collaborators, people that in surgery have really been helping us for this intraoperative work, and just want to thank them. And if people haven't seen our website, that's a place where you can find out more of what we do. So thanks for your attention. I know it was rushed, but I'm around and happy to answer questions now or later as well. Any questions? Thanks. Yes? Yeah, the costs. So some of these ophthalmology systems are in the range of maybe $50,000 to $80,000. The catheter-based systems for the coronary arteries are about $100,000. Those are quite expensive. A lot of the handheld primary care type systems that we're looking at, as OCT becomes more and more developed, the cost of the components continues to fall. So you could build a system, an OCT system in the lab these days for probably $10,000. And I think that's going to continue to kind of fall. So we hope for the primary care, at least, to be in the range of maybe $15,000 to $20,000. A lot like in these primary care settings, groups of physicians will share like an EKG machine, and we can envision maybe they'll start by sharing an OCT machine, and that price point I think is pretty reasonable. So it's only going to get better. Yeah.
How do you decide if you wanted to use swept source OCT or spectral domain OCT?
Yeah, that's a good question too. You're right that for the most part everybody is doing either swept source or the spectral domain. For my point, we're very interested in ISAM, these computational methods, Doppler, spectroscopic. All of these require phase stability of the light because you want to reconstruct sort of the optical wavefront and then manipulate that. So that requires the acquisition of the data to be phase stable. And when I say phase stable, that means if that interferogram that you're collecting, as you scan across tissue, if it's homogeneous or if you scan across a mirror, those fringes should not move. Those fringes should stay in the same place. That means phase stability. The spectral domain, or using a spectrometer, gives you much better phase stability. A swept source tends to be a little bit faster. So if we really want to do, megahertz OCT scanning, the swept source is the only way to go. But that has a moving component, and that's going to disrupt your phase stability. So that really is the deciding point in my mind. In the spectral domain, we use these cameras, and those cameras are getting faster every day. So right now, these cameras are up to about 140 kilohertz. So you can acquire 140,000 A scans per second, which is quite fast, and still have that phase stability. So we tend to, in my group, we tend to favor those types of systems. Yep.
Spectroscopy with OCT. In spectral domain OCT, you encode your position with frequency. So is it possible to do both simultaneously?
It is, yeah. So there are these. I flew through it. There's one slide that shows this sort of time domain, spectral domain relationship. They are related, and so you can still collect the interferogram data and analyze that spectroscopically as you would in the time domain data. Okay, I think we need to just take a quick 5-minute break, and then we'll come back for our next one. Okay.